Hello everyone, welcome to my channel Biophil. So today I came up with part 5 portion of infection and disease series. Please watch the full video and please comment if you like this video and please share your opinion in comment box. And if you like this video, please subscribe the channel. Let's move on to the first slide of this video. So, as I included this chart in my previous videos also, today I am going to cover the red circled portion that is included in eukaryotic microorganism. So, protozoa is the topic of this video. Let's move on to next slide. First is introduction. First, this term is parasitism. We should know about the parasitism thing. A parasite is defined as an organism that lives in a more or less close association with another organism of a different species that is called host and derives sustenance from it, and it is pathogenic to the host. So, this total feature of this organism is known as, and the relationship is known as parasitism. This extended process results in significant host parasite interaction at the cellular and organismal level. For example, Giardia lamblia induce biochemical change in the host cells at the site of infection, that is, the duodenal epithelium. Parasites cause numerous diseases in humans, some being of extraordinary significance. Example, malaria. So, we all know about the disease is malaria. So, protozoa is responsible for this disease. Let's move on to next slide. So, we will discuss in this slide some general features of parasitic protozoa. So, protozoa are eukaryotic single-celled microorganism. All pathogenic protozoa are heterotroph. Obviously, they show parasitism, so they must be heterotrophs. That means they are dependent to their host cells. Their cell size ranges from 1 to 150 micrometer and enclosed by trilaminated cell membrane. That means their cell membrane has triple layer possessing unique organelles such as kinetoplast, hydroxynosome, mitosomes, and all these organelles are involved in their unique metabolism. They have flagella, cilia or amoeboid movement for their motility. That means from one place to go another place they need this extracellular appendages, and they have this like flagella, cilia, and their reproduction mode is asexual or sexual. So we will discuss their habitats. When life cycles involves more than one host organism, physiology and morphology of the parasite will produce a significant change in the as a result of environmental change. That means whenever a protozoa has more than one host organism, their physiology and their morphological changes must be there due to their environmental changes, because hosts are different. So, inside the host environment is also different. That is why their morphologies are changed time to time, such as parasites moving from gut to other tissues. For example, it will encounter higher level of oxygen, changes in pH, and significant exposure to host immune response are greater. That means if some parasites first place is gut, and after that whenever they shift from gut to other tissues, they will encounter some changes, like oxygenic concentration is different in gut and the other tissue, deeper tissues, and same as for pH also, pH is also different, and immune responses are greater in tissues. The reasons for such movement include evasion of host immune attack and to aid transmission. That means this movement, that means this shifting, there should be some such reason. So what is the reason? So reasons are like they move from gut to tissue or from one host to another host is to protect themselves from host immune attack, and for their disease transmission also. Some species produce resistant stages like cyst, oocyst, in which the parasites can survive outside their host in longer period. That means in parasitism, our actual thinking is like that protozoal target of, that means their habitat must be inside the host, but in some parasites they show some survivable stages, some resistant stages like cysts. So these stages help them to survive outside the host for longer period. These are all protective structure of protozoa. This is a rough figure that shows the life cycle of some protozoa. See, first, first it is transferred from water or food material to human body, and after that it comes out from human body and remain as cyst, and after a specific time interval they can infect other organism, that means another host, or they can remain as cyst and again involved in disease causing other human being. So this is the common feature of their life cycle. So some species undergo nuclear division in cystic stage. So cysts are forms required to survive outside the host, with which nucleus giving rise to new trophozoid, that means the growing motile and pathogenic form. That means, see, trophozoid are the pathogenic form of protozoa. Okay. So here in human body, see, it's written trophozoid. So this is the pathogenic form of protozoa. Obviously it is present inside the host, and whenever they came out from the host cell, they stay as, it takes as the cyst structure. Okay. So this is the dormant structure of protozoa outside the host cell, and this is a protective structure for them. It helps them to protect from outer environmental conditions. Okay, this table represents the classification of different types of protozoa. So I'll discuss some examples such as euglena, amoeba, Psilophora, Microspora. These are all examples of subphylum of protozoa, and it includes class, order, and some genus name. That means the examples such as Entamoeba, Toxoplasma, etc. So you can go through this table. I think you will get more information other than I informed you. So the host response to infection. Mechanisms to control parasitic protozoa are similar to those utilized for other infectious agents. So they can be divided into non-specific mechanism and specific mechanism involving immune system. That means parasitic infection can lead to two type of immune reaction or immune response. Obviously, the first one is non-specific and the second one is specific mechanism. The sickle cell hemoglobin trait and lack of Duffy factor on the erythrocyte surface make the red blood cell more resistant to invasion by plasmodium. That means if these things are present outside the red blood cell, it is a protective mechanism for human being not to get infected by plasmodium. Okay, this trait are commonly found in population from malaria endemic region. That means wherever endemic, where malaria is endemic in such region, these factors can be seen there only. A second example of non-specific factor is the presence of trypanolytic factor in serum of humans, with which confer the resistance to Trypanosoma brucei, although non-specific factor can play a key role in resistance, usually they work in conjunction with the host immune system. So this is another example of non-specific factor that helps to get protected from Trypanosoma brucei and the disease caused by Trypanosoma brucei. So these are some examples of anti-protozoal drugs that helps to protect us from the disease or the extensive effect of protozoal diseased condition such as dapsone. So their unique target is cofactor synthesis of plasmodium species. That means they block the synthesis of cofactors of enzyme in plasmodium species. So there are other examples like metronidazole, quinine, then tetracycline, amphotericin B, pyramquine. These are all, they have different mode of action, and the target and their actual target is to stop the extensive effect of protozoal infection. These are the reference I used for this video. If you want to get more information, you can go through this, and please comment after watching this video. Thanks for watching this video. I hope this will be a helpful step for you. Thank you so much from my side.